I want to thank Mahmoud and the BioRTC team in Yobe for this invitation to give this talk. And I also want to congratulate them and the Yobe State University for this privilege of starting this center, which we hope will contribute as a research hub in Nigeria. I am going to speak basically on how we need to prioritize areas of comparative advantage for African neuroscience. I have just 15 minutes and I want to keep to the time. So when I'm talking about African neuroscience, for the sake of this talk, I am talking about basic neuroscience. So I'll be talking mainly about basic neuroscience for this talk to explain areas of advantages. So why do we have to talk about comparative advantage? Why is comparative advantage a big issue for African neuroscience? Well, first and foremost, particularly in basic neuroscience, is that there's low funding for research. So you do not have sufficient money for research work. There is no enough recurrent capital to do research. So obviously you do not have the luxury to spread yourself too wide because you will spread yourself too thin. The absence of funding also leads to low research equipment. So that's why we are happy for what is now happening in Yobe State University with this BioRTC center that is being set up, which can help in making sure that we have at least some basic equipment to work with. We're also talking of comparative advantage for African neuroscience because there are some unique flora and fauna that are found in Africa that have importance in neuroscience. So Professor Mate talked about some of unique plants that have been known to have anti-cancer effects. The literature in the neuroscience communication paper by Mahmoud et al. Just recently, so that there's increasing publication from Africa on medicinal plants and its role in neuroscience, and many being published in high impact factor journals. There are also unique diseases in the tropics that are not well funded and affect millions of people. So some of those infectious diseases, some of them are unique to the tropics. I'll be talking about that. You also have problem with lack of enforcement of regulation mandates. So makes in Africa, people exposed to some levels of pollution in some areas that you may not get in other parts of the world, and which has effects on the nervous system, on cognition. And lastly, comparative advantage. Even though we talk about comparative advantage, we are talking of things we can, like when Yorube was talking in clinical neuroscience, with conflicts, drug addiction, so things that have global relevance, but yet a local problem. So this can be a cut across, but this is why comparative advantage is an issue. So I give this overview in basic research on areas of comparative advantage, medicinal plants, environmental neurotoxicology, neuroscience of unique wildlife, stem cells involving indigenous animals, and infectious diseases that are unique to the African continent that affects the brain. So these are areas in which, particularly those who are starting off in neuroscience or who are trying to establish neuroscience laboratories, be it structural, morphology, behavioral, biochemical, molecular labs, can work on based on these areas of comparative advantage to the African environment. So the first one is medicinal plants. So like I said, Professor Mate talked about neuro-oncology. So there are plants that are known to be neuroprotective. There are plants that are known to aid memory. There are plants that are known to reduce oxidative stress. There are plants that are known to reduce aging degeneration. While on the other side of the coin, there are indigenous plants that are neurotoxic. There are indigenous plants that have that have potentials for addiction. So away from the cannabis that we are used to, local people have knowledge of some local plants that have addictive tendencies and sometimes also can be neurotoxic. So we need to have see this as a comparative advantage because some of those plants are completely unique to this environment. However, the problem, and which is why we are speaking now, particularly when a BioRTC center is being started and many young people across border are listening, is the science has to be done properly. So we need to move beyond the science of just taking a plant, squeezing the juice, giving it to the animal in the water, and then just saying this plant has caused this effect and that effect. So we need to move higher so that the science we do in our comparative advantage is yet good science, and then we provide better information to the research community. I have two papers recently involved in the lab, one in 2018. So this is the popular work from Mayowa on discovering or isolating the novel compound from moringa. So when everybody says moringa has this property, that property, but okay, moringa was taken, we did a methanolic extract, fractionation, liquid fractionation, separated into different fractions, tested each fraction for its antioxidative properties, used FRAP, and then selected few fractions and then compared them with standard controls, went into cell culture with those fractions, did Comet assay, looked at its protection in using immortal hippocampal cells. And then when we found one fraction that gave the optimal neuroprotective effect, so we tried to find out the compounds in that fraction, then using the nuclear magnetic resonance, did a characterization of the structure of that in that fraction, and then we're able to see the structures. And then we now synthesized, now that we now know the structure, synthesized the compound, and then now tested it in models of neurotoxicity to see its neuroprotective effect. So that's areas we need to go to so that we can bring out the great advantages, because the problem is if you are using plants and you say they have effects, you don't know the content. So when there is toxicity, even from the plant, you can't have an antidote because you do not know the compounds that are inside the, and likewise also, many times the active ingredients can now be synthesized and then can now be produced in a far more efficient way. And when it's synthesized, and then a modification of the compound can be done to have more neurotropic effect. So probably in some areas, it has an hydroxyl group. Then we need to synthesize a compound, and instead of an hydroxyl group, you need a phenolic, turn it to a more phenolic compound, or an oxyl group instead of an hydroxyl. So Modifications can be made to the compound to make it better. That is the advantage of isolating compounds, that structures in compounds. And in the right, this was a recent work from collaboration between Nigeria and Cameroon of a plant that is known to have both brain active effect and reproductive effects. So this was a folklore plant, and that is collaboration, and worked in our lab, and we were able to get the extract that could produce the neuroprotective effect and also protection against ovarian damage. And now we are moving now to characterization of active of the active ingredients within that extract that is producing this effect. So that's the way to go on medicinal plants. So from Cameroon to Nigeria to Ghana, so you see this increase in medicinal plant, but we need to do good science. So my next point is environmental neurotoxicity. That's also an area that we have problems in Africa, basically because many times there is lack of enforcement of regulatory issues, and then people live in around very toxic environments, be it from local mining, illegal mining of metals, of gold, and you have some of these gases from illegal mining into the environment, from dump sites, from crude oil burning, from crude oil extraction, from gas flaring, and many of those compounds affect whole communities. And we are, as neuroscientists, we have opportunities to even do basic research to find out the effect of these compounds on the nervous system. And apart from finding their effect, pathways that are being perturbed. So sometimes when we do basic research, the active compounds that are being released from the dump site or from crude oil emission, we can mimic those experiments in the laboratory, maybe through inhalatory route or other routes, and then find out whether it's microglia activation you have, or you have damage of neurons or astrocytic activation, and then with using Western blot or PCR, whether you have increased expression of COX-2, IL-6, which genes are downregulated, and is this short term? Is this long term? So these are good science we can do based on environmental neurotoxicology. And if we go back to what I said about the neurotoxic plants, if you match that with environmental neurotoxicity, so even when you have neurotoxicity, we can actually know plants within our environment that can aid those communities to at least mitigate the neurotoxic effects that they have. So moving quickly, you also have unique wildlife in Africa. I have you can see the picture here of Pomenja, who actually goes into the wild and across the continent, trying to study brains and neuroanatomy of unique wildlife. You see the brain there of the lion. Pomenja is working on the elephants. So we have some of these unique species in Africa that we can study, not just the histoarchitecture, but also gives us opportunity, neurophysiologists, wildlife physiologists, to study behavior of these animals in the wild through computer, telegraphy, and so that we can study their behaviors, and then in an event of death, study unique anatomy, unique structures in the brain. There are some of these unique neurons that we know from rats, locus coeruleus, orexin expressing neurons. So what are the expression of those neurons in African jackal, in the African elephant, in the giraffe? So some of these unique wildlife, we have an opportunity of studying unique neuronal populations within some of these unique wildlife. So these are areas that are still begging for attention, so that we can properly understand some of the functions of some of these unique neuronal populations by studying them also in this wildlife. So still on that same vein of unique neuroscience of African population is now also developing stem cells from some of these unique African rodents. So we are in collaboration and working on 2 species currently, the African giant rat and the greater king rat, to develop develop and then characterize neural stem cells from these species. Yeah, because the species have their unique ecology, they have their unique behavior, which has been studied over years. So harping on that basic knowledge that we know about them, we can also go into stem cells. So when you have the stem cells, you characterize them. It's easy to compare those stem cells with what you already have in the data bank of the sequence of microglia, genomics between some of these unique African rodents with that of the laboratory mice, with that of the laboratory rats. And we can now probably from evolutionary biology, evolutionary neuroscience, see how closer some of their unique molecular characterization is closer to humans. Let's know that animals like the greater cane rat is pregnant for 150 days, okay? So the laboratory rat is for 28 days. So there are unique developmental milestones within some of these animals that may make us better understand developmental neuroscience, in which we can contribute more to the neuroscience literature and understanding by studying some of these unique rodents. This is the African giant rat, and this is the greater cane rat. And here we are showing one of the latest papers that we have from developmental neuroscience. So this is the greater cane rat, in which we've mapped out developmental procedures. Like the animal is pregnant for 150 days, but gross brain development actually starts at about 50 to 60 days. So that's when the brain starts to actually develop. And that's when we even first, about day 40, about day 50 is when we saw the development of the neural tube. So it's easy now to map developmental milestones in a rat that is pregnant for 150 days. So things like, when do you have cerebellar development? When do you have the suprachiasmatic nucleus being developed? You have longer milestones here compared to what you have in the mice, who is developed in 28 days. So when you work with unique rodents like that, and that's why, apart from having a longer gestation period, it is also relatively more gyrencephalic. So genes that govern gyrencephalization, which you cannot properly study in mice, can be properly studied in unique models like this. And we've done some work with collaboration, actually looked at cortical development of these rodents at different time points, and look at unique developmental milestones, okay, with PAC6, with TBR2. These are unique genes that are important in development of the cortex. So with this, we have actually laid a good groundwork to move into stem cell, neural stem cell production in these rodents. So, and lastly, one other unique area for comparative advantage for African neuroscience is infectious diseases. So diseases that have been studied and have been relatively neglected, but remains a problem. Diseases like trypanosomiasis. So they remain problems in Africa, both human trypanosomiasis and animal trypanosomiasis. So these are areas we can still have a lot to offer by working in Africa, since we have access to the animals, access to the people, particularly even our clinicians, cognitive neuroscience and basic science, okay, to look at effects of trips, and with science now developing, trypanosomiasis being a sleeping sickness, to start looking at probably in cattle, in sheep, the effects that these parasites have on circadian rhythm genes, clock, timeless, and to see what they do to actually the genes of the time clock. And in this regard, like the moderator said, we recently got a grant from the Alexander von Humboldt Humboldt Research Hub for Africa. So they are setting up 6 hubs, and we are looking at zoonotic arboviral diseases. And I'll be particularly specializing on diseases viral diseases that affect the nervous system, okay? West Nile and Japanese equine encephalitis and such diseases. So these are unique diseases, viruses that affect the nervous system. And a lot of that is present within the African setting, and want to look through some of the West African to Central African countries. And for some of these very brilliant students, we are still particularly looking for master students who want to come to University of Ibadan to do masters, either in neuroscience or virology, that can join us in this huge work that we have to do. It's a 5-year grant, so particularly looking for master students. So I think with that, I have spent the time allotted to me talking about comparative area. I'll end with this. Comparative advantage is not enough. We build on comparative advantage to make sure that we do proper science. If proper science is not done, then the comparative advantage is not taken care of. So we need to make sure, like I told you about medicinal plants, let's get the extract, let's fractionate, let's test the different fractions, let's find out, isolate the active fractions, characterize the active compounds. So that is where we take good advantage of comparative advantage. If we do good science, what that leads to is that we have the opportunity to publish in high impact factor journals. So that means your comparative advantage done with good science can actually now get the attention of the world. So by publishing, Yarube showed good example there with some of those publications in clinical neuroscience, in pathophysiology. And once you start getting your publications in high impact factor journals, then you put yourself in good stead to write good research grants, okay, to have access to good grants. Grants are limited. Grants are not endless. So it's always given to the qualified. So but once you have good papers from in impact factor journal, it puts you in good stead to write good grants. And we also, the comparative advantage we have needs to be sold to the world. And that's where the issue of attending conferences, high impact conferences, IBRO conference, ISN, Society for Neuroscience, African Society of American Society of Neurochemistry. So top conferences that now bring, I mean, if we have unique rodents, we have their neuroscience, we have a good world stage to show our work. And we also make sure we are in active collaboration. There's definitely resource limited areas. We don't have enough funding, but if we do good science, we publish, we put ourselves in good stead for good collaboration to work with those who have good funding. So equipment, laser ablation, spectrophotometry, equipment, confocal microscopy, and electroencephalography, things that patch clamping that we may not have, but since we have a good idea based coming from comparative advantage of neuroscience, it puts us in good stead to have active collaborators in centers of excellence in which we can. And that's, it now becomes a vicious cycle of scientific prosperity, not a vicious cycle of poverty, because the active collaboration will lead to better impact factor papers, which leads to better writing of better grants, and also puts us in better world stage in top conferences. And that also leads to more collaboration, and then the cycle continues. So with that, I want to again thank Mahmoud for this milestone in what is happening in Yobe State, and hope that insurgency will end soon, and so that this can be good advantage for active science for people across the region. Thank you very much, Takeshi.